Thanks very much. We've had some great talks today, also today, hearing of how we're integrating in a multidisciplinary way our ways of tackling the threat of antimicrobial resistance. So I'm going to talk about something completely different, and that's the growing concept of using bacteria actually to treat or prevent bacterial infection. And this is not anything new. If you go into hospital for your third episode of Clostridium difficile diarrhea, the treatment you might get is fecal transplantation, which is feces from another person inoculated into your duodenum. And this is a very successful way of treating difficult clostid disease. This is basically altering the microbiome of the gut using bacteria from a donor. In the future, we'll be able to do this in a very much more targeted and directed way, using single organisms genetically transformed to express a desirable epitope within the gut. And this shows a paper published recently in Cell, where they influence the concentrations of autoinducer proteins in the gut in a mouse model, just with a single transformed E. coli, and completely altered the microbiome of the gut following antibiotic use. So in the future, we may be able, instead of putting feces into our patients' stomachs, we might be able to have them swallow a specific probiotic organism that's been genetically engineered. Likewise, the nasopharynx also has a huge microbiome. And in fact, colonization of the nasopharynx is the first step in a range of diseases. So, for example, in order to get pneumonia with strep pyogenes, you first of all have to be colonized in the nasopharynx by strep pyogenes and have a nasopharyngeal microbiome that's permissive to that. Likewise, you won't get meningococcal meningitis unless you've been recently colonized by Neisseria meningitidis. Now, my group was one of the first in the world to inoculate humans with live bacteria in the upper respiratory tract. And the organism that we chose to do this with is Neisseria lactamica, because this is completely harmless and in fact colonizes babies. Adults don't carry it. Instead, their microbiome is permissive to carriage of Neisseria meningitidis, which causes meningitis. And what we found, and this is a young clinical investigator who's still doing this kind of work, who inoculated some student volunteers with Neisseria lactamica, and what we saw was that this organism was carried in a proportion of those students for up to 6 months, quite harmlessly. And furthermore, it generated an immune response here in saliva, IgA, and also in the bloodstream, we detected IgG. So we were able to induce carriage completely safely with this commensal and make the host make an immune response to that organism. And we extended this work and published this just recently, showing that when we inoculated this harmless commensal into the noses of volunteers, in this case students, what we found was a change in Neisseria meningitidis capture. Now, as you know, students get meningitis. They get meningococcal meningitis at a higher frequency compared to the rest of the population. And certainly during the first term, because of social interaction, there's a lot of capture of Neisseria meningitidis in the nasopharynx. And what this slide shows on the y-axis is the rate of colonization in the throat of the meningitis bacteria, comparing those whom we inoculated with Neisseria lactamica versus controls. And you can see that there's a difference there. And what we found was that there was a significant drop in the acquisition of Neisseria meningitidis in students in whom we induced carriage of lactamica. So how are we taking this forward? Well, the idea is that we could indeed alter the microbiome of the upper respiratory tract in sophisticated ways using perhaps genetically modified, very safe commensal bacteria like Neisseria lactamica. But of course, before we can do that, we have to show that this system works and is very safe. We don't want to liberate organisms that, for example, carry nice antibiotic resistance markers. We want to use organisms that, when they're genetically transformed, are completely safe. So what we've done is to invent a system of transforming this organism at the chromosomal level, not using plasmids, because plasmids are transmissible. And one of the investigators in my lab, Dr. Jay Laver, has invented a system of doing something very difficult, which is putting into the chromosome of Neisseria lactamica bespoke cassettes that can express different antigens on the surface. And what's more, he's managed to do this without using antibiotic selection markers, instead using the endogenous beta-galactosidase gene that this organism has. And this is what he's ended up with, which is a Neisseria lactamica organism which expresses two molecules on its surface that it didn't before, and these are natural adhesins. In other words, they're molecules that enable a bug to colonize very well. And he's shown that these are expressed on the surface by all of the conventional techniques. And we've now been funded by the Medical Research Council to show that this system is safe. We haven't generated an organism that is more virulent. The organism does not change in its antimicrobial resistance during passage. And during 2016, we'll make a couple of applications. If you're going to do an experimental human infection, as this would be a proof of principle of that, and you want to do it in people that you let out into the community, you have to get regulatory permission to do that. So first of all, we're going to show that the organism is safe, and then we're going to have to apply to DEFRA to perform a deliberate release experiment under highly controlled conditions, and of course to the National Research Ethics Service to enable us to do the experiment in human volunteers. So I'd just like to acknowledge the help of the NIHR Biomedical Research Unit, who helped us during the early stages of this this work, and who will fund us to do the initial pilot study. Also grateful to the MRC, who have funded the safety studies that we now need to do before we apply to DEFRA and NRES, and of course my collaborators and my team. Thank you.